So here we're looking at the chemical synapse. So this is basically just the junction between two neurons or where two nerves meet. So here what you're looking at, you have the presynaptic neuron. So the presynaptic neuron is going to be this first one here that we're looking at, the top one. And then you have the postsynaptic neuron. So the postsynaptic neuron, you can just call it the second neuron, and then the presynaptic neuron, the first neuron, just to keep it simple. So what happens? You have this action potential that comes into the presynaptic neuron, and that action potential is going to be a voltage change. So it changes the voltage of this neuron. So because it changes the voltage, we have these calcium channels that are on the side of the actual neuron. They're going to be voltage gated calcium channels. And because this change in voltage is coming into the cell, it actually opens these channels. And you can see the actual potential coming in on both sides. Opens both the or all four of these calcium channels. Then you get this influx of calcium, which is just the calcium coming into the cell. So once this calcium, or you get this influx of calcium inside of the cell, what happens is you're going to have these synaptic vesicles start to move towards the terminal, the axon terminal, which is just this side of the axon, the end of it. So what these synaptic vesicles are carrying, they're carrying neurotransmitters. Now, depending upon what neurotransmitter it is, they could be excitatory or inhibitory in response. But here I just drew this so it could be a basic, just so you can understand what's happening. So once these synaptic vesicles move towards the axon terminal, they're made up of the same molecules as the actual membrane. So they're actually able to fuse with the membrane, and then through exocytosis, you have all of these neurotransmitters being dumped into the synaptic cleft. So the synaptic cleft is this area right here, or it's literally the junction or the synapse. So it's literally where the two neurons are meeting. So on the other side, or the postsynaptic neuron, what you're gonna see happen is that you have ligand gated ion channels on the outside or on the actual membrane. So all of these channels are ligand gated, and the ligand that they're binding is going to be the neurotransmitter. So once it gets dumped into the synapse, again, depending upon if it's excitatory or inhibitory, like if it's excitatory, it might be opening calcium and sodium channels, so it's allowing these ions to come into the cell. It's just going to push it towards threshold, and then it's going to allow the action potential to be excitatory, or the response to be excitatory. If it's an inhibitory response, they could be opening potassium channels where they're actually allowing potassium ions to go out of the cell, or they could be allowing chloride ions to come into the cell. Now, that's going to take it further away from the threshold for its local potential, and it's going to make it an inhibitory response where the neuron doesn't fire. So it just depends on what neurotransmitter is being released, but that's the basis of really what's going on at a chemical synapse.